My father was a translator. He spoke 5 languages fluently, and he was pretty good at lots of others as well. I used to marvel when I was a child, we'd go on holiday to different countries. He could converse with different people, whatever the land we were in. They could understand him, he could understand them. Sadly, I didn't inherit his linguistic skills. But methods of communication have remained with me as a fascination throughout my life. What about messages? What do they convey? How do we decipher them? How do we act on the information that we get? Can this translate not just into humans, but into other things, other forms of life? I was always interested in science, and in particular, the science of the human body and how the body responds to threats from the environment. How do we protect ourselves from infection? What are the cells that are used in protecting the body from infection? The cells of the immune system. So I was lucky enough to become an immunologist, and now I spend my time studying those cells, studying how they respond to infectious threats and other threats in the environment. And what I've learned is a key component of how the immune system responds is the communication between the cells. The cells are speaking to each other, and that's how we manifest our defense of the body. Let's think about the lungs. I've become very interested in the immune system in the lungs. Now, I hope you're all still breathing. Yeah, we haven't lost anybody yet. That's good. Every time you breathe, you're breathing in and out about 1/2 liter of air. That equates to about 10,000 liters of air that pass through our lungs every day. Everything in the air, we're breathing. And let's think about the challenges that poses to the lungs. And in particular, I want to tell you about a group of cells called epithelial cells. These are cells that form the barrier in the lungs between the body, the lungs themselves, and the external environment, the air that we're breathing. And if you think about it, they have a very, very challenging job to do because, yes, we want to keep out noxious particles, viruses, and bacteria, so they have to be able to do that. But also, they have to allow us to exchange the gases that we need for life. Every time we breathe in, we're taking a little oxygen from the air we breathe, and we're returning a bit of carbon dioxide. So these barrier cells are actually very useful to us, these epithelial cells that act as barriers. They allow us to exchange gases, and they must protect us. They must keep the outside out and the inside in. And for a long time, that's all we thought they did. We just thought they were barriers, that they didn't have much other functioning. Now we know, of course, that they do. And in fact, a key part of how they protect us from the outside environment, how they maintain the integrity of the lungs, how they keep us free from infection, is that they communicate with other cells. And some of the important cells that they communicate with are called macrophages. That literally means cells that go around eating things. They're big eaters. And the epithelial cells tell the macrophages what's in the environment. They send signals. They communicate with the environment. They send signals to the macrophages so that if there's an infection coming, if there's a bacteria that they've encountered, for example, the macrophages become activated to deal with that threat. And we call these activated macrophages M1. As immunologists, we're not very imaginative. So these are M1, and they're very aggressive. They're spoiling for a fight. They'll kick the hell out of bacteria. But of course, they might damage the surrounding. Just like in a bar brawl, lots of windows get smashed. So they take out the invaders, but they can cause damage. So we have to limit their aggression. And so the barrier cells also have to send signals to macrophages to calm them down. And in their calmed state, their chilled out state, they're known as M2. Again, very imaginative. So the M2 macrophages are laid back, and what they do is they stop the M1 macrophages from being aggressive. And the M2 macrophages go in and they repair any damage that's done. They repair that barrier, those epithelial cells. Clearly, the signaling between the epithelial cells and the macrophages is very important. And sometimes the macrophages return the favor. They signal back to the epithelial cells, as we shall see a bit later on. They can instruct the epithelial cells to do things. But this communication between the cells regulates the activity, and it keeps us healthy. It keeps our lungs healthy. As we breathe in everything in the air. Particles of soot, bacteria, people coughing, viruses, diesel particles, organic matter. Everything in the air that we're breathing passes into our lungs. And of course, that can cause damage to the lungs. If we know how the damage is caused, and perhaps which pollutants are causing that damage, then maybe we can do something about it. Now, the World Health Organization tell us that in the past year, over 7 million people have died from air pollution. That's about 1 in 8 of the total deaths in the world is due to breathing polluted and contaminated air. And that's a problem in our country, and particularly in many of the developing countries around the world. Pollutants can corrupt the signaling between those cells. Pollutants very often can activate the M1, those aggressive macrophages, and keep them switched on, so we get lots of damage. As a result, of course, we can get chronic diseases, asthma, COPD, chronic obstructive pulmonary disease, and many others that cause the deaths that we see due to air pollution. Other pollutants can activate the M2 macrophages and keep them switched on. And what can happen then is we get aberrant thickening of the epithelial cells, and we might leave ourselves open to infection from bacteria and from viruses. If we can understand those signals, if we can understand the language between the epithelial cells and the macrophages, and how the pollutants can corrupt that language, then maybe we can start to do something about it. And that's the task that I set myself and my research group. Now, the first thing to do, of course, is to find some lungs. We want to work on this signaling between epithelial cells, barrier cells in the lungs, and immune cells, the macrophages that interact with them. So I'd like volunteers to donate some of their lungs. It's all in a good cause. In science, yeah, I usually get that response. Not too many people want to give me some of their lungs. Actually, there are some people that do, and there's a technique called bronchoalveolar lavage, where you put a salt solution into the lungs, and you wash out the lungs, and with that comes some cells, and we can grow the cells. But we don't really want to do that. It's a bit of an inconvenience, and I wouldn't recommend that you volunteer for that, quite frankly. What we've done is develop cells that grow continuously, that are actually very, very much like the alveolar epithelial cells and macrophages, the cells of the lung that we want to study. We can grow these cells together in special chambers in the lab, so they're like a lab lung. So now we have a system that we can interrogate. We can put different pollutants into that system, and that's what we're doing. We can add extracts from diesel exhaust particles. We can add allergens. We can add organic matter that's come from composting or intensive agriculture sites. And that's exactly what we're doing. And we're looking at the responses that the macrophages have. Are they aggressive? Are they chilled out? Are they M1? Are they M2? What's happening to the epithelial cells? We can study all these things in this lab lung that we've got. Now, the cells don't talk to each other using words. It would be great if they did. but they talk to each other by producing small proteins. Those proteins are called cytokines. So if we can identify those proteins, that would be like identifying the language, and then we could translate what they were saying. We could understand the language of the cells. So that's what we need to do, and that's what we've been doing. We've been taking samples from our lab lung, our cell cultures, that have had different stimulants added to them, different pollutants, and we've taken the mixture containing all the proteins that these cells are communicating to each other with, and we're trying to identify what they are. Sometimes the macrophages talk back to the epithelial cells, and if there are particular stresses in the environment, the macrophages instruct the epithelial cells to commit suicide. It's a process called apoptosis. Now, sometimes this can be actually beneficial, but of course many times it can be detrimental. And if you look at the difference between the pictures, the micrographs that you can see on the slides, you can see that on the left you've got a nice mixture of macrophages and epithelial cells, but the main difference is on the right that many of those cells have disappeared. Those are the epithelial cells that have committed suicide under instruction from the macrophages. So we're interested in what those signals are as well. What's that language? Okay, to understand a language, we need a translator. Just much like when we started, I told you about my father translating languages. We have to have a translator that can understand the protein language that the cells are speaking to each other. We have to be able to take all the proteins in our system, put it through a translator, and hopefully some sensible sentences might come out the other end. And to do that, we use sophisticated equipment called a mass spectrometer. What this does is it smashes up all the proteins into tiny fragments, and then it separates those fragments according to their mass. And it turns out that the mass of the fragments are unique to different proteins. So if we can identify the fragments from their masses, we can work back and tell which proteins are present. And that's exactly what we're doing. We're looking at all the proteins in our system, something called proteomics. We're analyzing that and then deciphering what proteins are produced under different conditions, what pollutants are inducing certain proteins, and what's the response from our macrophages. So this is the language of the cells. This is the protein language of the cells. It turns out the protein language is quite complex, but we're starting to understand it. We've got a rudimentary understanding of the language of the cells. And this will allow us to make predictions about how the cells might respond in different situations. So what have we found out? We've been able to grow lung cells in the lab. We've been able to add different stimulants, different pollutants to these lung cells. And we've been able to look at whether they induce the aggressive M1 macrophages or the chilled out M2 macrophages, and how that impacts on the barrier cells. We've been able to find out some of the protein language that instructs the aggressive M1 macrophage or the chilled out M2 macrophage. And this is allowing us to develop drugs that can maybe change that language so that when we're faced with pollutants or other stresses that switch on the aggressive M1, we can change that language, we can modify it back to a language that tells them to chill out a bit. So we might be able to treat or even prevent diseases such as asthma and other airway diseases, the more we understand about this language, this protein language of the cells. And more than that, our mini lung is also, we can work the language back the other way. We can look at what it's telling us and then understand what's in the environment around us, what is in the air around us. So we're using our cells as sensors to tell us what's around every breath we take. So please think about the language of the cells with every breath you take. Thank you.